Good afternoon to everybody. First of all, thanks for attending this webinar, the second of many, according to our commitment to sharing knowledge. Let me first introduce myself and my company. My name is Ruth Ordóñez, and I am a research and development engineer and bioprocessist specialist in Bionet. After finishing my PhD in chemical engineering, with which I acquired real experience on mammalian filtration, I joined Bionet in 2011 to create the filtration department. From here, I take care of processing support to customers on downstream issues. At the same time, I develop basic and detailed engineering of new filtration projects. At Bionet, we are manufacturer of laboratory, pilot, and industrial equipment, and of software focused to bioreactors, CIP units, tangential flow filtration, as well as complete bioproduction plants. There, we integrate our proprietary equipment with technologies from other leading vendors like centrifuges and homogenizers. Briefly, the content of the webinar is divided into an introduction, a second chapter for presenting the main type of membranes, a third section for showing the parameters used for characterizing the feed solution, followed by a guide for selecting most suitable membrane module and the final main conclusions. I don't want to finish the webinar without showing you an example of one of the plants we have developed in Bionet for vaccines production. And the last but not least, we will also have time for questions in the platform. Let's start from the beginning. Before understanding how TFF is related to vaccines production, let's see the main vaccines that exist. Vaccines are necessary for preventing viral and bacterial diseases, and they can be inactivated and live but attenuated. Attenuated vaccines are based on a virus or bacteria strain that has been weakened in order to decrease its virulence. This type of microorganisms can replicate in the body but are not pathogenic. Inactivated vaccines contain the pathogen inactivated or killed to eliminate infectivity. These vaccines can be made from the whole pathogen cell or from subunits, like the ones made from toxins or the ones called virus-like particles, which are specific vaccines that mimics the overall structure of virus without the infection's genetic material. There is a new generation of vaccines derived from genetic engineering, like the ones we can see in the screen. Some of them will be commented later. Filtration implies installing a porous membrane between two solutions. By applying pressure at one of the membrane sites, the components are transported toward the membrane surface. There, some of them will pass through it, producing the permeate fraction, while the rest will be retained. We can set 4 different types of barriers. Microfiltration, it is the less restrictive, and it only retains suspended solids like whole cells and cell debris. Pore size is in the range of 0.1 and 0.8 microns. Normally, it is not necessary to apply more than 2 bars to filter. Ultrafiltration, we use it to retain macromolecules like proteins or DNA. Pore size goes from the N of mf down to 1 kilodalton, and it may require pressures up to 5 bar. Nanofiltration, with pore sizes down to 200 Dalton, it allows to retain even multivalent ions after applying up to 20 bar. Finally, reverse osmosis, which only allows the pass of water, and it has been reported pressures up to 120 bar. MF and UF are based on physical screening, while the NF and RO are also affected by the osmotic pressure of the media, acquiring importance the interaction solute-water membrane. Most bacteria are between 0.6 and 4 microns in diameter, so they can be seen with an optical microscope, while viruses, notably smaller, can only be detected by electron microscopy. In that sense, microfiltration would be enough to retain bacteria, but in case of virus, UF is absolutely necessary. Tangential flow filtration, or TFF, is a type of filtration where the starting solution is fed in parallel to membrane surface, where implies the following advantages. Certain fraction sweeps along particles, what has a surface cleaning effect, allowing a permeate flow more stable over the time. More or less, all the vaccines share the same production chain, which includes an upstream, downstream, and final formulation. The upstream involves the production of the vaccine. Production can be done from different sources that include eggs, cell culture, insect cell culture, and microbial fermentation. Get vaccine from eggs is the most ancient method. The principle is to inoculate embryonated chicken eggs with the virus of interest, which then replicates in the chorioallontonic membrane. Recently, several viral vaccines have moved away from an egg-based process and have adopted a cell culture procedure. The primary reason for this shift is to prevent vaccine production issues associated with embryonated eggs, like, for example, a possible shortage of egg supply in the event of any avian disease outbreak. In mammalian cell culture, the virus is grown in the mammalian cells, being the most common the cold May-Darby caning kidney, or MDCK. The virus infects the cells that express 4 viruses extracellularly. Baculovirus are a diverse group of DNA viruses that can infect insects in nature but are not pathogenic for humans, so they are perfect tools for use them like vaccine vectors. A viral vector is a modified virus that acts as a vehicle to introduce exogenous genetic material into the nucleus of a cell. Insect cells are cultivated to a desired cell density and then are infected with a recombinant baculovirus containing the gene of interest. Microbial fermentation is used, for example, in the hepatitis B vaccine through recombinant DNA. First of all, the virus antigen-producing gene is isolated from the raw virus. At the same time, a plasmid DNA is extracted from a bacteria and is cut with restriction enzyme forming the plasmid vector. The isolated virus antigen-producing gene is inserted into the bacterial plasmid vector, forming the recombinant DNA. This recombinant DNA containing the target gene is introduced into a yeast cell, forming the recombinant yeast. Recombinant yeast cells multiply in the fermenter and produce the virus antigens. The target compound used for formulating the vaccine can be found extracellular, like the one used for the meningococcal or typhoid vaccines, and intracellular, like the rotavirus, virus-like particles, or viral vectors. Once the culture or fermentation is finished, the product has to be harvested, entering into the downstream area. In the case of intracellular compounds, a lysis step has to be included before clarification in order to break the cell and release the target molecule. TFF is the best alternative to phase clarification, concentration, and purification steps. Finally, the formulation section collects several steps with the final target of preserving and stabilizing the vaccine and to potentiate the immune response. Diverse technologies are used for clarification, including centrifugation and filtration technologies like tangential and direct filtration. Several series of operations are often required to achieve desired clarification. The first operation, called primary clarification, is aimed at removing larger particle-like whole cells. The second one, called secondary clarification, is used for removing colloids and other submicron particles. Centrifugation, as a choice for primary clarification, enables cells and cell debris removal by sedimentation. It can handle high solid load, and it has been extensively used in batch or continuous mode with this stack centrifuge. However, it requires high capital investment and maintenance costs, and it presents challenges related to scale-up. Direct filtration, applied through depth filters, retains particles by size exclusion, and as they contain positively charged material, it enhances retention of cell debris, colloids, and negatively charged unwanted components. They are easily scalable and implementable. However, they are not of high holding capacity. Therefore, they are more recommended for secondary clarification to avoid their blockage. TFF, however, is linearly scalable. It can face a high solid load, and the membranes are reusable after cleaning, so it can be implemented for both primary and secondary clarification. The clarification step is sometimes neglected during vaccine process development because time and resources are more focused on production and purification steps. However, clarification efficiency directly influences the performance of the downstream process. A poorly optimized clarification step can negatively impact the capacity of sterilizing grade filters or may shorten a chromatography resin lifetime. For all these reasons, clarification step must not be a bottleneck. In this context, TFF is living up to the new clarification challenge, addressing increased process flexibility, possibility of single use, and reduced investment costs. TFF is also a perfect friend after clarification. In concentration, is applied to isolate the product of interest in the lowest possible volume. In this case, the desired fraction is the retentate generated after filtering in the UF range. In purification, is also used to reduce insoluble impurities, host cells, proteins, or host cell nucleic acids. In this case, the desired fraction is also the retentate generated after filtering in the UF range. Let's see now an example of production train for the influenza or flu vaccine. Influenza virus is an enveloped single-stranded RNA virus that causes the influenza, what we all call the flu. There is no template process for cell culture based in influenza vaccine, and manufacturers follow different methods of manufacturing and select various technologies with their own process philosophy. However, we can set the following typical culture-based flu vaccine process. First of all, a sterile cell culture media has to be prepared in order to set the perfect environment for cell growth. Secondly, the specific flu cell strain has to be inoculated into the media to cultivate it until achieve cell concentrations around million cells per milliliter. Between 72 and 96 hours after seeding, cultures are infected with influenza virus strains. After incubation of 3 to 5 days, host cell culture can produce up to 1,000 million of cells per milliliter that express the virus extracellularly. After this moment, we can proceed with the harvest. During primary and secondary clarification, host cells and cell debris are separated from the virus of interest by tangential flow filtration, first by 0.45 microns and then by 0.22 microns. At the end, the virus is recovered in the permeate fraction. The next step pretends virus inactivation by formaldehyde addition or by UV radiation. After inactivation, whole virus particles are concentrated in order to reduce the process volume downstream. This concentration can be also done by TFF. UF combined with DF, or diafiltration, is performed to remove low molecular weight impurities like protein and host cell DNA or for buffer exchange. A more selective purification step by chromatography is normally necessary to remove the impurities that still remain after filtration. Most flu vaccines are split vaccines, which are produced by detergent treatment. During this step, the structure of influenza virus is dissociated by breaking down the envelope and releasing the internal antigenic components. Virus splitting removes some of the viral components, resulting in less reactogenic vaccine. In order to guarantee sterilization before formulation, direct filtration through 0.22 microns is necessary. Final formulation requires the addition of adjuvants or other components to improve the immune response to vaccine antigens. The main type of membranes modules used for vaccines production are ceramic, hollow fibers, and cassettes. Ceramics have the advantage of being resistant to solvent and to extreme pH. They present a long lifetime, whenever they are cleaned properly, and they can handle solutions with high viscosity and suspended solids content. On the contrary, they are not compact. The cost per square meter is high. They are fragile, their use implies high energy consumption, and they are not gentle with the product. Ceramics are multitubular modules. As more channels, more filtration area, but the number of channels and their diameter is determined by the viscosity and the content of solids of the raw solution. The main material for these membranes are titanium dioxide and aluminum. Hollow fiber membranes are also multitubular, but with tiny channels which diameter and length depends on the viscosity and the content of solids in the feed solution. In this case, they are more compact and cheaper per square meter than ceramics, and they handle the product very gently. However, compared to ceramic, they are less tolerant to suspended solids. Their lifetime is lower, and they are more limited to solvents. Regarding materials, in this case, they are organic and highly hydrophilic for guaranteeing low protein binding. Cassettes are more recommended for UF applications as they can manage high protein concentrations. They present higher fluxes than hollow fibers and they are really compact. On the contrary, they are not gentle with the product and their cost per square meter is really high. They are also made of highly hydrophilic organic polymers, and in parallel to ceramics and hollow fibers, the thickness of their spacers depends on viscosity and solid content. In order to select the most suitable membrane module, we need first to characterize the solution where the target product is contained. Pore size and material selection is affected by the biochemical composition of feed solution and the biochemistry of the target molecule. It is not the same to clarify a lysis fraction, which has a lot of underside compounds interacting with our desired component, than clarify an extracellular component. It is also important to have in mind the amount of feeding product in order to filter it in the feasible time. The amount of final product is also important in order to control the dead volume of the system during design. Another important parameter is the temperature. Filtration should be done at the highest temperature acceptable for the membrane and for the product in order to maximize filtrability. Viscosity and/or cell density also affects on membrane module and pumps to select. Finally, shear rate sensitivity is another key parameter to have in mind. Some biomolecules exhibit low mechanical resistance and may be damaged by shear exposure. So, this issue will not only impact the type of membrane module to choose, but also the pumping technology. The manner we have for manipulating shear stress is through the cross-flow velocity in ceramic membranes, through the shear rate in the hollow fibers, and by the cross-flow feed in cassettes. As higher these values are, the more stress is applied to biomolecules. With all the previous information shared, we have all the tools on the table to create a guide for membrane selection. The choice of membrane pore size is important for minimizing filter fouling and product losses. Fouling will shorten the time for which a membrane can be used before it must be cleaned. In general, membranes with smaller pores will show less tendency to fouling because particulate material cannot penetrate and block the pores. Then we have to make ourselves three main questions. Is our product shear sensitive? What is the working pH of my solution? What is the viscosity of our solution? The correct answer to this question will give us the recommendation about the best membrane module to use. In case that our product is sensitive to shear, hollow fiber will be our preference, but limiting the shear rate to 2000 and 4000. If our product is not shear sensitive, we could choose any membrane module. In terms of pH, if it is extreme, ceramic will be the recommendation, but for normal pH, any membrane module could be used. If, after answering to a previous question, we still don't know which membrane use, viscosity value could be our key. Whenever viscosity is above 60 centipoise, ceramic is the most recommended option, but taking into account the channel diameter in order to work under acceptable pressure drop values. In case that viscosity is below 60 centipoise, any membrane can be selected. For hollow fibers, we need to consider both membrane length and lumen diameter, and for cassettes, the screen thickness has to be considered instead. In a future webinar, we will show you the selection tools applied in practical experience with real broths. I have just showed you how to select a membrane module from a theoretical point of view. However, sometimes it's not clear what is the best membrane to apply. In all that cases, the best solution is to compare all of them. In Bionet, we have developed the M1, a unique large-scale system for the technology screening, optimization, and scale-up activities. It provides unique flexibility because it can work with the membrane technologies in the MF and UF range. It allows testing membrane from the main leading vendors. They are completely interchangeable. And it can grow from basic manual to complete automatic and modular way. Normally, when we face a new filtration process, we distinguish two main steps. First of all, the technology selection, where we select the most suitable membrane module, as I have explained. Then, we need to optimize our process, in which we determine the key performance indicators that define our filtration process. I'm talking about cross-flow velocity, TMP, permeate fluxes, concentration factors, etc. Both steps can be performed with the M1 system, and even if the filtrability of the product is acceptable, we could process small production batches below 20 liters. If you want to check all features about M1, you can access into our website. And to finish, some conclusions. We have seen that DFF is an effective tool for proceeding with clarification, concentration, and purification steps in the downstream of vaccines production. Main membranes that can be used for vaccines production are ceramic, hollow fibers, and cassettes. A correct pore size selection minimizes fouling and product losses. Selection of the most recommended membrane module is determined mainly by shear sensitivity, pH, and viscosity or cell density. The M1 unit is a unique solution for technology screening, process optimization, and scale-up activities. Here you have an example of one of the plants we have supplied for vaccines production in Bionet. This plant was built for the veterinarian industry. The vaccine was obtained with the whole cell produced from bacteria fermentation. As the target product was in the retentate, it was designed with a very low dead volume in order to achieve high concentration factors before formulation. Concentration was done with more than 2 square meters of ceramic membranes. The plant was completely automatic, not only for operation but also for SIP and CIP sequences. I want to mention that the design was made to keep an sterile filtration, not only in the recirculation tank, but also in the membranes. As I said at the beginning, there is going to be some time for questions. We will be online for a while, answering them and publishing the ones that we think are relevant. Please, when making a question, leave your email so that we can answer them and provide you with supporting information or material. I'm delighted to invite you to the next webinar also about principle of turbidostat and how to add precision to your continuous process strategies, held by my colleague, Dr. Juan Moreno Cid. We will publish date and time in the coming updates. Thanks for attending this webinar. I'm really happy sharing this moment with you, and I hope you find it useful. We wait you in our social networks like Facebook or LinkedIn, in our website, and here's an email in case you want more information. If you want to be updated about all the Bionet activities like webinar schedule, enter into our website and register for the newsletter. And finally, on behalf of all my Bionet colleagues and Bionet, thanks for your attention.